We'll record that too. And we'll share the screen. All right, so where are we at now? We're on week 13. So we're trucking right along. As we come to week 13, where are we at? Today's the 26th, 27th, yeah. So don't forget, exam 4 is obviously not due by the 18th. It's due by May 2nd. So May 2nd would be this Saturday. Just a quick reminder. And then in lecture today, we're going to begin our investigation into the nervous system. And we're going to be covering chapter 11, probably for all of today's lecture and maybe even all of next week. Chapter 11, there's a lot to talk about with the nervous system. So just hang tight in there. Don't forget, if you're trying to follow along, you can print out the Ivy Learn notes. So it's no fun in telling you this again, but I always like to make sure everyone's on the same page. Today's lecture, we will be covering chapter 11, the central nervous system and the peripheral nervous system. So if you'd like, you can download that, and that'll kind of go along with today's lecture, give you an idea of everything that you're responsible for. All right, so exam 4 is this week. It's up on Ivy Learn, and now we have our chapter 11 lecture for today. Any questions before we begin? Not too bad. Let me find my little chat box. Yes, absolutely. What's up? So next week is our last class. Next week is our last lecture meeting. Yes. So I will post, what is it? The week after is when the exam is due. But yeah, I'm not actually going to put any new material on it for obvious reasons. It's our final exam. Now, just a reminder that the final exam is half comprehensive and half over chapter 11. So everything that we've talked about up to this point in lecture, you will be responsible for. So half comprehensive and over half chapter 11. I'm almost certain I won't get into chapter 12, so I'm going to just cross that off. I'll cover a little bit of that in lab, but we'll keep that with the lab material. Now, if you're trying to figure out what to study for the final exam, if we go to our homepage, I don't know if I have something for the final up here or not yet. If not, I do. I was going to say if I didn't put it up there yet, I will post it, but there it is. So if you download that study guide, this will tell you everything that you're responsible for on the comprehensive portion. So there are a lot of important things you guys want to know as you move along, such as your different type of organic molecules, different electrolytes, the concepts of pH and things like that. So I did put that up there for you guys to follow. Now, as for the final exam, just to give you guys a heads up, it's just like the other tests. It's 60 questions, 120 points. So there's no big final exam. It's just 60 question test. 120 points. So what I'll probably do is pull 30 questions out of chapter 11 and then 30 questions out of the comprehensive material, or I just may just make up new ones. You never know. But they will follow along with that study guide. Sound good? Well, if you have any other questions, comments, suggestions, don't hesitate to ask those. Add those in the chat box. Remember that shortcut, Alt-H. I wonder if it works. I know. What a long journey. What a long, strange trip it's been. Let's just say that. But and unfortunately, our summer classes will also be virtual, and I won't be surprised if fall classes also begin to be virtual. I think we're going to be in this for a while, so we'll see how that turns out. All right, guys. Well, with that said, let's turn our attention to chapter 11, and we're going to talk today about the nervous system, specifically the central nervous system. All right, so we're going to begin, and today we're all going to be looking at the central nervous system. I will go ahead and tell you that the textbook, if you guys are using the 15th edition, they've rearranged some of the chapters. So it's not some of the chapters, but some of the sections. So it's in a little bit different order, but it's still the same material. It's just not, they cover the brain first and then the spinal cord. I like to cover the spinal cord first, but you'll see why hopefully later on. All right. So as we begin looking at the nervous system and the central nervous system in particular, the first thing we want to know is that it develops from a very simple tube, which we call a neural tube. So the first thing that we're going to look at is how we actually develop this brain from a sheet of tissue that starts out as the neural tube. And what you're going to see is that this neural tube actually begins as a flat plate, as a sheet of tissue. And this is the neural plate. And what we'll see here is that these edges will start to fold up like this, and they will enclose the tube. And so now we have this groove going right down the center, and eventually these two sides will come together, and this will completely close the tube. And if we're looking at the tube, something like this, it is not a solid tube, it is rather a fluid-filled tube. So in the middle, we have this cavity. And this cavity is filled with cerebrospinal fluid. So now eventually the top part of this neural tube will give rise to the brain, whereas the bottom gives rise to the spinal cord. So brain and the spinal cord. And the fluid-filled cavities, as you guys might guess, will eventually become the ventricles at the level of the brain. We talked a little bit about those in lab. And in the spinal cord, it will then give rise to the central canal. Central canal. All right. And this is how we begin the development of the central nervous system. Now, it's imperative that this tube closes, that the edges of the tube actually come together and seal to form this enclosure. And this is the dorsal side, so that would be along your back. Sometimes though, this doesn't happen. Sometimes part of it remains open. And if this is the case, then we develop what we call a neural tube defect. And depending at the level of the central nervous system, the defects can vary. If, for example, this occurs at the level of the brain up here, then the brain itself will not develop. You'll be missing either the entire brain or parts of the brain. And this is called anencephaly. So with anencephaly, what we see here is a neural tube defect at the level of the brain, in which the brain itself will not form. However, if the neural tube defect occurs down here in the spinal cord, well, the brain will develop just fine, but then we'll have some type of paralysis perhaps at the level of the spinal cord. And this is what we call spina bifida. So spina bifida is basically where this neural tube does not close. It remains open at the level of the spinal cord. Oh, I'm sorry to hear that. Yeah. Yeah, anencephaly, I should mention too, is a lethal thing. Yeah, you will often have a baby that will maybe survive for a very short time, but it will ultimately typically be a lethal condition. Now, spina bifida, this will either lead to paralysis or it could be asymptomatic. What that means is a lot of people will have it and they won't know they've had it until they go to the physician and they're doing a normal checkup and there's a mole on your back. I look at the mole like, what is this thing? Oh, that's when the neural tube did not close. So yeah, so asymptomatic means that you're not showing any types of symptoms. By the way, do you guys know what do they tell you to take during pregnancy or even before pregnancy to help prevent neural tube defects, specifically spina bifida? Folic acid. Now, there you go. Yeah, folic acid. So to prevent, you're going to take folic acid for prevention. Now, I always want to remind you guys that when you do take prenatal vitamins or vitamins in general, that you, of course, always want to follow a physician's prescription and don't just go take them all willy-nilly, because too much of the vitamins can definitely have their own detrimental effects. Vitamin A is the first thing that comes to mind, but definitely early on in pregnancy or even beforehand, folic acid is a good way to help prevent this. All right, now that the neural tube has formed, What happens is at the level of the brain, it starts to swell and it gives rise to three, what we call vesicles. And these are the primary vesicles. So the three primary vesicles are the three swellings that will ultimately give rise to the brain. And we'll start with the common names first, and then we'll go back and pick up the scientific names. The first area, this kind of big area here in blue, this is all called the forebrain. So when we think of this forebrain, that's that kind of teal area that we see at the front. Behind the forebrain, we have the midbrain. The midbrain is this kind of area there in pink. So first the forebrain, and then the midbrain. And as we come back, this green area is then called the hindbrain. So that's pretty easy to remember. It goes forebrain, midbrain, hindbrain. And of course, for anatomists, that's a little too easy to remember. So they give these long, fancy names for each one. For example, in the forebrain, we will call this the prosencephalon. It's basically the area before the brain. So here we see the prosencephalon. That is the forebrain. The midbrain, we will call the mesencephalon. That's the middle brain. So behind the forebrain, we then have our midbrain, the mesencephalon. And then this hindbrain here, this area in the back, this is called the rhombencephalon. I suppose where it's rhomboid shaped, but there is our rhombencephalon. So these are the three primary vesicles: prosencephalon, mesencephalon, rhombencephalon. From these three primary vesicles, they will then divide out to make 5 secondary vesicles. So just to show you how this is working, up here in the beginning, we start with the prosencephalon. So this whole area from here down to here is all the prosencephalon. And what you'll notice is that the outer part starts to swell. They form these nice big swellings. This is called the telencephalon. Down below, there's a little pouch that's also forming from the prosencephalon, which we will now call the diencephalon. So telencephalon, diencephalon, both of these are derived from the forebrain, or the prosencephalon. As for the mesencephalon, it doesn't divide into anything. It stays in the mesencephalon. So back in the midbrain, this area back here, mesencephalon is still the mesencephalon. As for the rhombencephalon, it divides into 2 secondary vesicles. So this hindbrain back here, this rhombencephalon, will then give rise to a metencephalon and then the myelencephalon. Of course, below the myelencephalon, you have the spinal cord. But there you have it, metencephalon and myelencephalon, both derived from the rhombencephalon. So the way this goes, telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon. Now, if you're trying to remember the order of the vesicles, here's a good way to do it. Here's how I like to do it. First, let's start with the area at the top. You know, do you guys remember Princess Diana? Very famous, what was she married? Prince Charles, I think. Yeah, and she had this horrific car accident when the paparazzi were chasing her. Oh, it was a big tragedy, yeah. 1996, wow. I didn't realize it was that long ago, or 97. Well, there you go, yeah. Well, here she is, Princess Diana, down here. I don't know what your princess looks like, but there's my Princess Diana. 97, there we go. And of course, the paparazzi were always after Princess Diana. You know what they did? They would always take these big telescopes and then use those telescopes to peer in to look at her. They were following around with these telescopes. So here we have some telescopes that are peeking down in on Princess Diana. So telencephalon for the telescopes, diencephalon where the telescopes are pointing. They're all pointing at Princess Diana. Now, the next three, the mesencephalon, metencephalon, and myelencephalon. Let me put them all over here on the same side so we can see this. Mesencephalon. They all occur in alphabetical order. So the way this goes, it would be mes, met, my. And that'll give you the order of the secondary vesicles. Do notice, by the way, that this myelin is spelled slightly different than your myelin sheath. So do keep that in mind. All right. With that said, let's see what they ultimately give rise to in the fetal brain. So this just shows us the development of the three vesicles. So here we start in the area of gray. This was the forebrain, which we call the prosencephalon. Here's the mesencephalon in yellow. And then this kind of pink area is the rhombencephalon, or the hindbrain. As we can see, these guys start to divide. So this prosencephalon gives rise to these nice big swellings on the top. That's the telencephalon. And then we have these smaller swellings down below, the diencephalon. The mesencephalon, it's still mesencephalon, so it remains the same throughout. And ultimately, we will go back to calling it its common name, the midbrain. As for the rhombencephalon, here we see it divide into a metencephalon in front, and then the myelencephalon behind it. All right, so here's the parts of the brain that they give rise to. If we were to take a section, the telencephalon will ultimately give rise to the cerebrum. So the cerebrum is that nice big outer part that you guys probably think of when you think of the brain. We have those gyri and the sulci. So that telencephalon will give rise to the cerebrum. As for the diencephalon, it gives rise to these areas in the center, the thalamus, the hypothalamus, and the pituitary gland. They put the epithalamus in there, but I'm not holding you responsible for that, so you don't need to know it. As for the mesencephalon, that of course gives rise to the midbrain, and I'll show you this in a second. As for the metencephalon, this gives rise to the pons on the front and the cerebellum on the back. And last but definitely not least, that myelencephalon gives rise to our medulla oblongata. So let me just show you these structures on the actual sagittal brain. And here's what we're looking at. So that telencephalon that we're looking at is giving rise to this whole area, the cerebrum. Check. The diencephalon gives rise to these smaller areas all down here. And we take a closer look. We'll just kind of zoom in. And we'll go into more detail of this later on, but there is your thalamus with the intermediate mass in the middle. There's your hypothalamus. And then you can see hanging down below that is the pituitary gland. So part of that pituitary gland, not the entire thing, but part of it does come from the diencephalon. Pituitary gland. And that's that area right there. All right. With that said, let me get rid of this. As we move back to the mesencephalon, I kind of colored this in red here because they labeled it in red down here. That will give rise to the midbrain. This is the first part of the brain stem that's connecting to the diencephalon. And then the metencephalon, like I mentioned before, it'll give rise to this pons towards the front. And in the back, it gives rise to the cerebellum. And last, but definitely not least, myelencephalon will give rise to the medulla oblongata. Your medulla oblongata. All right. Don't worry, guys. I'm going to go through these parts of the brains over and over and over again. Hopefully, you will know them by the back of your hand by the end. But definitely keep them in mind because we will start following the passageway as we connect this to the spinal cord. So far, okay? All right. Well then, guys, before we get to the brain, before we get to the spinal cord, we should also remember that during the development, we had this layer of connective tissue called the meninges. Awesome. Surrounding it. So let me go ahead and put up the meninges here. There we go. And of course, the meninges is not just one layer of tissue, it's actually composed of 3 separate layers. And so this is basically surrounding your brain and the spinal cord. So by the way, this area out here, you can see this is the gray matter. If you guys remember, the gray matter is the part of the central nervous system that is unmyelinated. And we're going to start with the innermost layer first. And the innermost layer is this, what we call pia mater. Pia mater is that gentle mother. That's this layer of connective tissue that sits right on the surface of the brain and the spinal cord. And it follows all these little convolutions and these grooves right there. If you're going to do a brain dissection, whenever you see, I wish I kept one. I should have put a picture in, but that's okay. There are these little blood vessels that cover the surface of the brain, and they're all part of the pia mater. It's very delicate, and it's a vascular layer of connective tissue. The next layer out is the arachnoid mater. And arachnoid, hopefully you guys are starting to think of a spider. And I'll show you why here in a second. Actually, you can see it right here. But here's the arachnoid mater. Notice that the arachnoid mater is not connected to the pia mater. It is separated. There is that space, what we call the subarachnoid space. So parts of the pia mater are actually connected to the arachnoid mater by these ligaments. And these ligaments here give it this kind of web-like appearance. But there is the arachnoid mater. Now that space in between the two, this is what we call the subarachnoid right there. And then last but definitely not least, the dura mater. The dura mater is that tough mother, the outermost layer. So here's our dura mater, dense layer of connective tissue, and it should sit right on top of the arachnoid mater. So there we have it, guys. Dura mater, arachnoid mater, the subarachnoid space with the CSF, and last but definitely not least, the pia mater. And that would be the meninges. By the way, do you guys know what they call inflammation of the meninges? Meningitis. You ever hear of meningitis? Yeah, there you go. Absolutely. Meningitis. That's quite okay. Oh, yeah, it's a real nasty thing. Oh, man. I'll tell you what. I don't know if you guys remember this. It's probably been about maybe 10 years ago now. There is a steroid company that makes these injections, that makes these steroids for people's, like often for back pain, thing like that. And so you'll get this steroid injection in the back, and it's supposed to help strengthen the muscles up, to help alleviate the pain, to help speed up the healing. And unfortunately, the steroids were contaminated with a fungus. And these people that would get injections would get this fungal meningitis. And it, I think, infected like 200 people. It was hundreds of people. It was many, many people. Several of them died from it. But yeah, it's definitely nothing to mess with. So meningitis is the inflammation of the meninges. All right, guys. Let me see. I think I have another picture of the meninges. Yeah, this is something else I want to show you. What I want to show you here, I don't know if we can see it, but this just gives us another view of the meninges. And what we can see here is that the pia mater is right on the surface of the brain, like this. So there's the pia mater. I'll just make it like that. There's pia mater as well. And here is the arachnoid mater, separated from the pia mater. So this is our arachnoid mater here. Now you'll notice that some of this arachnoid mater will come up and protrude through the dura mater. I'll talk about this later on. These are called arachnoid granulations, but we'll see those later on. So there is the arachnoid mater right there. And the thing I want to point out when we get to the dura mater itself, in some areas will separate into two spaces. So we have one part of the dura mater that goes right with the arachnoid mater like this. So these two are held together. That's the dura mater. But however, in some areas, it goes right up next to the bone. And this is what we would call the periosteal dura. So there's part that's on the periosteum. And here is the part that's on the meninges, what we call meningeal dura. Now, I'm not concerned about that, but I want you to notice is that there is a fluid-filled space right here that opens up in between these two. This is called a dural sinus. And it's these dural sinuses that will ultimately drain the CSF. It drains it back into your blood supply. So don't think that the cerebrospinal fluid is just a static pool. It's actually a continuous flow. And so these dural sinuses here will ultimately drain the blood, or not the blood, but the CSF into the bloodstream. I'll come back to those in a little bit, but that just shows it's a nice little area right there where you have the dural sinus between the meninges and the periosteum of the bone. All right. Now, with that said, let's move on inside the central nervous system and talk about the ventricles. And the ventricles are these fluid-filled spaces, these openings that we see throughout the central nervous system. And here we kind of see in pink these two what we call lateral ventricles. We used to have what we call a first and second, but they no longer label them like that. For now, we all just call them lateral ventricles. So you'd have one in the left hemisphere over there and a second one in the right hemisphere over here. Now these two lateral ventricles, they will then empty into a third ventricle. And this is what we see right there, ventricle number 3, occupying the mid-sagittal plane of the diencephalon. So we're going to see this later on, but that's at the area of the diencephalon. Now to get to the fourth ventricle, there is a narrow channel called the cerebral aqueduct. So you don't really see it here because the lateral ventricles come blocking it, but this channel that's going right down there, that would be that cerebral aqueduct that allows the CSF to flow from the third to the fourth ventricle. And last but definitely not least, our fourth ventricle is right there in the brainstem. Sits just behind the pons and in front of the cerebellum. So yeah, anterior to cerebellum. And there we have it, guys, the ventricles. Now, to understand how they're filled with CSF and how the CSF is produced, we have these kind of cauliflower-shaped capillary beds inside the ventricles called the choroid plexus. I'll come back to this in a second. So if you look, for example, up here, these little cauliflowers right there, these are what we call a choroid plexus. And it's the choroid plexus that actually secretes the CSF. There's a couple in the lateral ventricles. There's one on the roof of the third. And here's another one down here in the fourth ventricle. So these guys are secreting the CSF. And what we'll see, if we look at this from the side, choroid plexuses would be here and here. And then, of course, we have a little more on the third ventricle and some more in the fourth ventricle. We'll put them right there. And what happens is they're secreting the CSF. As they secrete the CSF will fill up this ventricle here and ultimately move inwards to come to that third ventricle. From the third ventricle, it goes down to the cerebral aqueduct. So there's our little chamber or cerebral aqueduct where it comes into the fourth ventricle. From the fourth ventricle, some of this will go outside into the subarachnoid space, while some of it continues down through the central canal. So this is actually going to be connecting to that subarachnoid space of the meninges. And what we're going to see is that as it comes down, the subarachnoid space here and the central canal here, it'll eventually work its way back up. And as it comes up towards the top, then that CSF will ultimately drain into your dural sinus. And this just shows you a nice area where we see one of the sinuses that's draining the CSF. So that's the whole idea with the CSF. Now, as for its function, its main purpose, it does a lot of different things. Number one, it's going to provide buoyancy. What this means is that if you take your brain, your spinal cord, and if you held your brain in your hand, if it was just sitting there in your hand, it would weigh about 3 pounds. It's a pretty heavy organ. But you know what? If you put it in this, and you set it in a floating pool so it could just float there, that would reduce the weight to about 1/2 pound. And so what this CSF does is it protects the central nervous system. It makes it in its own isolated pool. And it protects it from shearing forces So as you're moving back and forth, your brain's and your spinal cord is going to be moving inside. But fortunately, that CSF helps to cushion that. In addition, I had some other functions. I must have erased them. That's okay. Let me put some major functions of the CSF. Another important thing that it's doing is maintaining the electrolyte balance. The electrolytes in your CSF are different than your the rest of your plasma. So it maintains electrolyte, pH, nutrient balance, in addition to providing that buoyancy. Also, it removes waste. You know, as you go throughout the day, neurotransmitters are getting broken down, molecules are being broken down, parts of cells might starting to be releasing their debris. You want to get rid of that debris. And so thankfully to this nice slow circulation, this will help get rid of it. Interesting side note, this is, by the way, one of the hypotheses of sleeping. You ever wonder why sleep is so important? If you can figure this out, you'd earn yourself a Nobel Prize, I guarantee it, because this is one of our greatest mysteries in biology, why sleep is so important. But for right now, one idea, and the one I'm sure has many functions, but what we notice is that during sleep, the circulation of the CSF starts to increase, and the sinuses, the spaces start to increase. And we think that this extra flow while we're sleeping helps to remove the debris that accumulates throughout the day. So very lovely thought out there, but you know, I always like to put it out there. Excuse me. All right, one other thing I would want to mention, if you're following the central nervous system down, you're going to notice that the spinal cord itself terminates at the second lumbar vertebra. So there's L1, there's L2. It's usually right in between those. And there we can see the end of the spinal cord. However, the meninges continues down. So the dura mater and the arachnoid mater will continue on down to about the second sacral vertebra. So this might be your dura mater. And you're going to notice there's a sac here that's filled with CSF. This is called the thecal sac, also known as a dural sac. And this is important because this dural sac has CSF. Hey, there you go. Yeah, just like the band, Spinal Tap. No, I'm kidding. Yeah, so if you want to get a sample of the CSF, or you may want to monitor the pressure, you may want to look to see if there's any blood in there, or you may want to administer medication, then you can put a needle right here, typically between the L3 and the L4 vertebra, or you may do it between the L4 and the L5 down here. And what that'll do is it'll give you a sample of the CSF without puncturing the spinal cord. That way you don't have to worry about paralyzing somebody. So here you can see they're just kind of bent over in this position to help extrude those, to make those vertebrae more prominent and easier to find. Now, by the way, a lumbar puncture or a spinal tap, this is different than an epidural, but they are in very similar areas. So this just shows us that a lumbar puncture is actually going to go into the subarachnoid space. So here would be our subarachnoid space with the CSF. An epidural, if you want to administer this, you're going to go into a space just above the dura mater. So this is on the outside. You're going to see it's just in the same area, just not quite as deep. But you might, I'm sure you've heard of an epidural. That's where you're going for this one. A spinal tap, you're going to go just a little bit deeper so you can get to the CSF that's in here. Well then, guys, that does it for our meninges and our ventricles. So let's now turn our attention to the spinal cord. And what we're going to do is start with the spinal cord, take a look at its anatomy, and then we'll just kind of work our way through the major tracks, the things that are going through the spinal cord. The first thing you'll notice as we look at the spinal cord, it's not just a nice slender tube. It actually has a couple of prominent swellings. First, it starts up at the hole in the base of your cranium. You guys remember the foramen magnum? Yeah. Foramen magnum. And of course, if you're above the foramen magnum, we're talking about the brain. If you're below the foramen magnum, then we're talking spinal cord. As it emerges from the foramen magnum, it gives rise to a cervical enlargement. So this would be up by your neck area here. And then here is our thoracic region. And then down in the lower back, it gives rise to a lumbar enlargement down here. And then it ends at about the second lumbar vertebra. So there's L2. There's a little structure we call the conus medullaris. I'll introduce that in a second. But that's the end of the spinal cord. This area down here, the spinal terminal, this is actually connective tissue. It's technically not part of the nervous tissue. So we're going to leave it alone for right now. By the way, whenever you look at the spinal cord, it's hard to appreciate, but to give it approximate size, it's about the size of my pinky finger or your pinky finger. So it's actually rather small. And in fact, it's very delicate too. If I was to do this, that probably would have just decimated that connective or that nervous tissue. It would probably would have just crushed it. So you can imagine it's very important to protect this area of the central nervous system. So thankfully we have those meninges. Thankfully we have that CSF to help protect it. All right. So this just shows us our conus medullaris, the C2. I'm sorry, not C2. The L2 end right about there where the spinal cord ends. And like I mentioned before, the filum terminal, that's connective tissue that comes down, anchoring it to the base down here. Now, I mentioned the cauda equina before. The cauda equina are actually spinal nerves. So to show you how this is coming off the spinal cord, let's go back. We'll use this one. Let's use this. Just a reminder, as we're looking at this, let me move it. Oh, I know what I want to do. Move this out of the way. There we go. As we're looking at the spinal nerves, put it like this. They are typically coming off the spinal cord like this. So here you have your cervical nerves. Here you'd have your thoracic. Down here you'd have your lumbar. Oh, a little higher. There you go. And your sacral. But as you get down here, some of these spinal nerves that come off will start to come down like this. And as they come down like this, they're going to form this kind of tail-like structure. So you're going to have these nerves that are hanging down below the spinal cord. That is what we were talking about with the cauda equina, a horse's tail. I'll show you a better picture of that, hopefully later on. All right. So cauda equina are these spinal nerves that are kind of fanning outwards down below the spinal cord itself. Now, as we begin looking at the spinal nerves, we like to number them by the level that they rise. So here's our different segments of the vertebral column with the spinal cord intact. The first area, as we start out here, is the cervical area. And here we have 8 pairs of cervical nerves, C1 through C8. Do be careful though, guys, because if you remember, how many cervical vertebrae do we have? About 7. Yeah. Remember, guys, we only have 7 cervical vertebrae. Oh, we have 8 nerves at 7 vertebrae. Yeah. So there, oops, let me go up here. Cervical vertebrae. Then we have 8 pairs cervical nerves. So the idea behind this, what's going on, is that there is an 8th nerve that comes out from below the C7th vertebrae. And so we label it C8 nerve. So do keep that in mind. And I'll revisit this next week probably. Thoracic level, we have 12 pairs of thoracic nerves. So fortunately, that's the same, T1 through T12. Same thing in the lumbar region. We have 5 pairs of lumbar vertebrae, L1 through L5. As you get down to the sacral region, here's the sacrum. You'd have S1 through S5. So there are 5 pairs of sacral nerves. And last but not least, all the way down to the coccyx, we have 1 pair of coccygeal nerves, which we just label CO. So if you add these up together, you get 8 plus 12 is 20, plus 5 is 30, 31 pairs of your spinal nerves. And I'll show you a little more detail of this as we begin looking at the spinal cord itself. So what we're going to do is just take a basic cross section through the spinal cord. And we're going to pull it out and take a look at it to see what's going on. All right, so here's our nice cross section of the spinal cord. The first thing you'll notice is that the spinal cord is divided into left and right sides. So if this is our anterior view, this would be the right side, and this would be the left. And what you'll notice is that there's a deep fissure running down the front. This is called the anterior median fissure. And that runs along the anterior side. On the back, there's a smaller depression called the posterior median sulcus. So if you're looking at your spinal cord, it'd be something like this, where the anterior median fissure goes in the front, and the posterior median sulcus goes in the back. And this will separate the spinal cord into the right and left sides. Notice in the middle, we have this central canal. So in the middle there, you'd have your central canal. And this is, of course, filled with cerebrospinal fluid. So in the middle of this, you're going to have your CSF. There. There it is. Now, it is useful to note that as we begin very small, the central canal is rather large. Then as we age, it becomes smaller and smaller and smaller. So depending on the sample that you get, it may either be kind of large or it may be rather small. All right, guys, now surrounding the central canal, we have this kind of butterfly-shaped pattern of gray matter. So do you remember, guys, that the gray matter is unmyelinated? So what we typically have in the gray matter are the cell bodies, the neurons, just like so. So all of this is gray matter, unmyelinated tissue. Now you'll notice that we divide the gray matter into horns. On the front, we have the anterior horn here. That's that nice big round area there. Off on the side, we have a lateral horn. Now, depending on where you're at, the lateral horn might be hard to see. It might be kind of small, or it might be more prominent, or it may not be existing at all. But here you can see the lateral horn off to the sides there and there, where the anterior horn is down there. And of course, in the back, we have the posterior horn back here. And here it would be on our spinal cord model. So anterior horn, lateral horns, posterior horns. And I'll talk more about the function of these guys later on. But in the meantime, you do want to realize that there is a bridge of gray matter that connects the right and the left sides. So for example, let's say we have a neuron over here and it's going to switch sides and it wants to communicate to another cell that's over here. And this other cell might continue to go upwards or something like that. Or a neuron on this side may cross over to this side. So we have this kind of bridge of gray matter. This is called the gray commissure. And the gray commissure, they've left it unlabeled here, so I'll go ahead and put it in, is an area where the neurons cross over. By the way, I'll talk more about this later on, but we give this a special name. Whenever neurons flip from one side to the other, we call this decussation. You know, crossing over. It's not like John Edwards crossing over. I don't know if you guys ever remember John Edwards. He was this really weird, pretend he could talk to the dead guy. People actually believe it. But anyway, decussation. So if you ever hear me talk about these neurons decussate, this is what I'm talking about. Now, surrounding the gray matter, we have these areas of white matter, and a white matter is, of course, myelinated. I don't want that. Let me get rid of this. I want that. There we go. So we divide these into columns because if you're looking at this, you can think of the white matter as these columns of axons that are basically coming up and down through the brain. They're going straight up like this as they ascend towards the brain. So there, it looks like maybe a nice big thick slice of pie. This area in the back is the posterior column. Off on the side, we have these lateral columns. And then in the front, we have the anterior columns. So each side would, of course, have two columns. You have one on the right side and one on the left side. So just to make things even, I'll go ahead and put another column up here like this. There you can see as it connects to the brain. By the way, another name for a column is also called a funiculus. You'll see later on why I call it a funiculus. Well, part of it is just fun to say. I mean, heck, the word fun is even in there. Look at that, fun. So, funiculus. Anyway, there you have it, guys. Anterior funiculus, lateral funiculus, and then back here you have your two posterior funiculus. All right, that's pretty much for the spinal cord right now. I want to move on to the spinal nerves, but first I want to talk a little bit about something of terminology. If you guys remember, I talked about this in lab, but I want to make sure everybody's on the same page. Depending on whether we're in the central nervous system or the peripheral nervous system, we use different terms based on our location. So remember, central nervous system, this is referring to the brain and the spinal cord. So anything inside the central nervous system, let's say this is your central nervous system. Here is your brain and the spinal cord. If you're in the central nervous system, whenever you have a group of cell bodies all clustered together, let's say they're all packed like this into a nice cluster, this is called a nucleus. So a nucleus, don't think of the nucleus of a cell, nucleus of an atom, but here we have a different type of nucleus. This nucleus is a cluster of cell bodies all together in the central nucleus or in the central nervous system. So just to give you an example, here's a label, what we call the nucleus accumbens. We'll talk more about this later on, but that's a nucleus, that's a group of cell bodies. If we have some axons all projecting together, let's say they're all connecting downwards like this, or maybe they're going to the front to the back, we call a bundle of axons a tract. So a tract is referring to a bundle of axons in the central nervous system. And here, this will remind, I'll talk about these later on, but coming out from the eyes, these would be what we call the optic tracts. These are major axons going from the eyes back to the visual cortex. All right, if you're outside the central nervous system, if you're in the peripheral nervous system, remember this would be your cranial nerves and your spinal nerves. In the peripheral nervous system, whenever you have a group of cell bodies all clustered together like this, we call it a ganglion. So a ganglion is a group of cell bodies all clustered together. Great example of this would be your dorsal root ganglion. It's a bunch of cell bodies all packed together in this swelling. And of course, connecting to these cell bodies, we have our axons. Whenever you have a bundle of axons all coming together like this, we will call this a nerve. So a nerve is where you have a bundle of axons plus their connective tissue wrappings all bundled together. So just to give you a couple examples here, here is the median nerve going down to your wrist and the ulnar nerve going down towards your pinky finger. All right, I mention this because I will start using different terms as we move along, and hopefully you'll start to understand where I'm talking about. So for example, if I talk about the dentate nucleus, hopefully if you hear the term nucleus, that would be a dead giveaway that we're in the central nervous system. It's a group of cell bodies. If I start talking about the spinothalamic tract, which I will, then you will also know that we're talking about these axons in the central nervous system. If I talk about a lacrimal ganglia, that's actually responsible for tears, you will notice it's a ganglion, so it's in the peripheral nervous system. And if I talk about the vagus nerve, well, hopefully since you hear that term nerve, you'll know it's in the peripheral nervous system. All right, guys, there's just a little bit of our definitions for us. Now, with that said, let's move on and talk a little bit about the spinal nerves. So as we start looking at the spinal nerve itself is just this little segment right here. It's very short, very small. But you'll notice that as it approaches the spinal cord, it bifurcates, it splits off into two. It gives rise to a ventral root, which is in the front. And the ventral root actually contains motor neurons. So here in the anterior horn, we have motor neurons, also known as efferent neurons. I don't know if I like to say efferent or efferent, but whatever you prefer. And their fibers will all project outwards going through the ventral root. So they go like this, and then they'll go to different muscles of the body. So maybe you have a skeletal muscle out here, like that. So the thing to keep in mind is that the ventral root is all purely motor. These are all motor neurons. Now the dorsal root's a little bit different because the cell bodies of the dorsal root are not actually in the central nervous system. They're in the peripheral nervous system. So if we look out here, you're going to see a swelling in the dorsal root called the dorsal root ganglia. And the dorsal root ganglion is referring to this nice big swelling right here. Dorsal root ganglion. I guess if I spell it out there, so you don't need to do it again, but DRG. Old habits die hard. And what we can see, if we look closely, is that it actually contains the cell bodies of the sensory neurons. So these little swellings right there and there, those represent the cell bodies. These, by the way, are what we call unipolar cells. I don't know if you guys remember the unipolar neurons versus the multipolar, but these are all unipolar. So to define this, the dorsal ganglion is a group of sensory neuron cell bodies. And we can see that their fibers are now all connecting. Maybe they're coming out here from some receptor. I don't know. Maybe it's from your fingertip. Let's say that's my fingertip. And you got to touch a receptor. This would be a sensory neuron with some dendrites out here. And that will be projecting inwards like this towards the central nervous system. So the thing to keep in mind is that the dorsal root contains nothing but sensory impulses. It's all sensory. Ventral root, all motor. Dorsal root is all sensory. So I mentioned this in lab, but it's worth going over again, but just way to think about it. Let's say you're doing a surgery, and during the surgery, you accidentally nick the ventral root. If you accidentally nick the ventral root, let's say up here, well, then that person's going to lose their motor output. Their muscles will become flaccid, they'll become weak, they may not be able to move a certain limb because you've cut the motor supply to that limb. If, on the other hand, you accidentally nick the dorsal root, then they're going to lose sensory. They might be able to move their hand okay, their limb okay, but they won't be able to feel certain things. They won't be able to feel pressure, pain, temperature, things like that. You can poke them, you can pinch them, they won't feel it. If, on the other hand, you damage the spinal nerve itself, then they're going to lose both sensory and motor. Spinal nerves are all mixed, right? They will become both flaccid and have weak tone, and they won't be able to feel touch. So spinal nerves are all mixed, which means they have sensory and motor. There is one exception. It's C1. Just for complete sake, I'm going to ignore it. C1's all motor, but the rest of them are sensory and motor. All right, guys. Now, in this next segment, what we want to do is talk a little bit about a reflex arc. So, as we start looking at these spinal cords, it's going to have two main functions. The first function that we'll talk about here first is having our spinal reflex. You guys are all familiar with this. You put your hand onto a hot surface. Let's say you put it over a candle flame like this, or maybe you accidentally touch it onto a stove. You don't even think about it. Hopefully, what's the first thing you're going to do? You're going to pull it back, right? That's what we call a withdrawal reflex. And these have to be super fast because you want to minimize the damage done to your nervous tissue. So fortunately for us, this does not even go up to the level of the brain. This occurs right at the level of the spinal cord. So as soon as that damage, the sensory from the damage comes in, it will trigger a muscle contraction and it will cause you to pull your arm back. And that's important. You don't want it to go all the way up to the brain. You don't want to sit there and think while your hand's on that hot flame or the hot stove, gee, my hand's starting to burn. What should I do? Should I just leave it here or should I pull it back? So that's the reflex arc. That's what we'll talk about first. The other thing to think about is that the spinal cord is also the main conduit for the nerve impulses going from the rest of the body to the brain, right? So the perception of pain, for example, when you put your hand on a hot stove, that's going to go up to the brain. That you will have to actually perceive. So with that said, guys, let's first turn attention to a reflex arc. And we can think of a reflex as a very simple response to a stimulus. So you all know the concept of a reflex arc. You have a sensory neuron that's bringing information in, and it will synapse onto a motor neuron with the information coming back out. Now, if the sensory neuron is directly connected to the motor neuron, this is what we call a monosynaptic reflex. Monosynaptic. And that's typically what it's not really typically what we'll be dealing with, but that's one example. It's just sensory to a motor. However, there are often what we call interneurons. Interneurons are the neurons completely in the central nervous system that serve to bridge those two together. And these interneurons can help the two communicate, and they can also help modulate your reflex arc. So for example, I don't know if you guys have ever tried to resist the urge to kick whenever the doctor taps your knee, or if you ever try to resist the urge to pull your hand back from that damaging stimulus. Yeah, so you can modulate these reflexes up to some degree, and that's thanks to these interneurons. All right, guys, I tell you what, let's go ahead and take a 5-minute break here, and then we'll come back and we'll talk more about these reflexes, and then I'll show you some major passageways through the spinal cord. So we'll do a little 5-minute break here, and then I'll see you guys back here in a few. There we go. Welcome back, class. So in this section, we're going to begin working our way through the spinal cord, starting with the basic concept of a reflex arc. And then from there, we're going to build on to more complex pathways as we begin to connect the spinal cord to the brain. So with that said, let me go ahead and get started right with our spinal cord. Let me find my pen there. There we go. And this just shows us the 5 basic concepts or the 5 basic components of the reflex arc. And what we're going to do is follow this in order. Now, the first part of any reflex or any stimulus is that what we call a receptor. A receptor is a part of the body that can detect a stimulus. So it could be like a photoreceptor, it could be a touch receptor, a pain receptor. All of these guys are able to detect a sensory stimulus, either a change in the body or a change in the environment, or body, because it could easily be the body as well. Now from here, we will transmit this information towards the spinal cord through a sensory neuron. So this cell right here going upwards into the spinal cord is a sensory neuron, also called an afferent neuron. Remember guys, sensory neurons, afferent neurons, they arrive to the spinal cord. Afferent, because they arrive. All right, that will then come back to that dorsal root. So there's a cell that would be in the dorsal root ganglion, and now here it comes in. Now in the central nervous system itself, you'll notice that connecting the sensory neuron to a motor neuron, we have another cell. This is the interneuron. So here is the sensory neuron in purple. This yellow cell that connects the sensory neuron to the motor neuron, that is the interneuron. So that's going to integrate the information in the central nervous system. So we go from the sensory neuron to the interneuron, the third part here, connecting the sensory to the motor. Then up here in the anterior horn, we have our motor neuron, and that motor neuron will come back out through the ventral root, and then it will send an impulse right to your muscle, and that will allow you to quickly pull that hand away from the stimulus. So these are the 5 basic concepts, the 5 components of the reflex arc: the receptor, sensory neuron, the interneuron, the efferent neuron or the motor neuron, and last but not least, the effector. Now the effector is what we think of as the muscle or a gland, right? You guys remember we talked about an effector. It's the thing that causes the effect. It's a muscle or a gland. In this case, it's the skeletal muscle that you'll be using to pull your hand back. So here we are, very similar thing to what we were just looking at. First, we have some stimulus. So for example, in the withdrawal reflex, we had that very hot flame that was detected by some type of pain receptor. From the receptor, we then go through a sensory neuron, through the dorsal root. From the sensory neuron, we connect it to an interneuron. Interneurons, if you guys remember, are completely inside the central nervous system. Inside the central nervous system. In this case, it's completely in the spinal cord. And then from here, we have our motor neuron. Motor neuron starting out the anterior horn. It will come out here and then synapse onto some effector, some muscle or gland. And last but definitely not least, you'll have your response to where you pull that away. And that's it. That's really the basic idea of a reflex arc. Now, some reflexes can be more complex, but for our purposes, this is a very clear demonstration of a typical reflex arc. You apply a stimulus that's detected by some sensory neurons, connecting in the central nervous system through interneurons. Then we project back out through the motor neurons and finally to the effector. All right. Now that we understand the basic concepts of a reflex arc, we're going to begin connecting the spinal cord to the brain. And to do this, you're going to notice that these axons that are connecting the two, they're projecting in 2 different directions. Some of these axons are starting down in the spinal cord, and they will be going upwards to the brain, bringing in their sensory information. This could be the sense of touch, pressure, pain, temperature, whatever it is. This tract, these are guys that are climbing, these are called ascending tracts. Ascending tracts are all sensory inputs. So it's containing sensory information going up to the brain. Other areas will begin in the brain. Maybe there's some cells up here in the cerebral cortex like this, in gray matter, and their axons will project downwards like this, eventually going out to either some type of muscle or to some gland. These are called descending tracts. Descending tracts are all motor outputs. So they will either synapse onto some muscle or some gland or connect to them ultimately. So whenever we start talking about these tracts, these are going to be located in the white matter of the spinal cord. If you guys remember the white matter, we divided it up into columns. We had the posterior columns, lateral columns, and the anterior columns. These are where we typically find the tracts. So we're going to begin by looking at some ascending tracts first, some sensory tracts. I'll talk about those important information, and then we'll come back and pick up some descending tracts afterwards. So remember, guys, with ascending tracts, these are all sensory input coming in from the body to the brain. Whoops. There we go. So these are all sensory. Now, the first two tracts that we'll look at occupy the entire posterior column. So here we have the posterior column on the right side and the posterior column on the left side. And back here, there are two important tracts. The first one we'll look at is called fasciculus gracilis. Fasciculus gracilis is a nice slender tract right along the midline back here. So there's one on the left side. Here's fasciculus gracilis on the right side. Now, whenever I talk about a fasciculus, that's just part of a funiculus. Gracilis refers to its slender nature. Sitting next to fasciculus gracilis, we then have fasciculus cuneatus. Cuneatus is referring to this wedge-shaped form like this. So this area right back here is fasciculus cuneatus on the left. Here's fasciculus cuneatus on the right. Both of these tracts are all relaying information regarding light touch from skin, also pressure, and proprioception. Now, I've not talked about proprioception yet, but here's the basic concept. You know, inside your muscles, there are special stretch receptors that are detecting their exact position. So I can close my eyes without even looking. I can hold my right hand up and I know exactly where my index finger is. In fact, I can take my pen and I can probably touch it to it right there. And that's because as I'm holding up my limbs, there are special stretch receptors throughout my arm, throughout my muscles, that are telling my brain exactly where that limb is. This is called proprioception. Think of it as a limb's position in space. All right, so touch, pressure, proprioception, all of these will be going through the posterior tract or the posterior columns. And they travel through these two tracts, fasciculus gracilis and fasciculus cuneatus. The only difference really between these two is that fasciculus gracilis is bringing the information in from the lower limbs, whereas cuneatus is coming from the upper limbs. And the reason this works is because if you imagine your foot, let me put a foot down here like this. And then up here we have the hand. Imagine as you're going up the body, you're going from the foot to the shin, to the thigh, to the hip. You're going to start by conveying that information in towards the midline like this. And as you climb higher and higher, all of this will start to go outwards like this. And that's going to go right towards the center, occupying that fasciculus gracilis. By the time you get to your upper limb, however, let's say your fingertips, now we're going to go out here towards the next column, fasciculus cuneatus. And what this does is it allows you to bring all the information in without having these crossing of the wires. They all go parallel with each other. All right. So these are those two tracks. We'll come back to this in a second. In the meantime, we're going to now cover the anterolateral system. So what we're going to see here is that unlike the posterior system, the anterior and the lateral system are really all bound together like this whole area. And so we're going to see these tracks just kind of overlapping. The first tract that we'll look at is called the spinothalamic tract. Now you'll see the spinothalamic tract goes all the way from the side to the front right here. So there's a lateral portion and the anterior portion of this tract. I'm not too concerned about the anterior portion. The anterior portion is also for touch, but it's more of a crude light touch. What I mean by that is with the posterior system back here, fasciculus, gracilis, and cuneatus, these are very fine touch. You know exactly, you know when somebody comes up and they poke you and you can feel exactly where they touched you. That'll be these systems back here. But sometimes maybe somebody just brushes up against you and you're not quite for sure where that touch is. That'll be the spinothalamic tract. But the main thing to keep in mind for this tract is the lateral side, because the lateral side is the one dealing with pain and temperature. So for example, you know, if you're getting ready for work this morning, or maybe you're getting ready for class, you want to take a nice hot shower and you turn the shower on, it's too hot and you don't realize it, and you jump in and it's burning and it's feeling hot pain, that's this guy right here. The lateral spinothalamic tract. Now I'll talk about these later on, but these fibers, as they're starting to come up, at some point they will all start to cross over. They will decussate. They will go from one side to the other, and then they will continue up. So for example, let's say I put my right hand on a hot stove. That pain will actually be going up to my left brain. Likewise, if I take my left hand, I put it on a hot stove, that will then go up to my right brain, because these sensory neurons are continually crossing over. Decussation. That's our crossing over. All right, one more tract we should take a look at called the spinocerebellar tract. So the spinocerebellar tract, we can just see it's all along this edge right here. I'm not actually concerned that you know where these are at, just know what they convey. That's the big thing. And here, the spinocerebellar tract is sending up the sensory for your body's position. It allows your cerebellum to monitor your body's position, where you're at, where you're needing to move. It's going to allow you to coordinate your movements. So for example, remember when I talked about proprioception? I can take my hands, I can bring them together like this. And that's because if you guys remember, the cerebellum is continually monitoring my position. It's keeping track of it and making this correction. That's what this tract will be doing. Spinocerebellar tract is the sensory for balance. All right, so there we have it. Our four main ascending tracts: fasciculus gracilis, cuneatus, spinothalamic tract, and spinocerebellar tract. Now what we're going to do is follow one of these tracks through the spinal cord up to the brain. But before we do, I do want to go ahead and mention the basic concepts, the anatomy of the brain. So just a reminder that this is the cerebrum. And you'll see this sulcus right here. That is, I don't know if you guys remember this from lab, but this is what I call the central sulcus. In front of the central sulcus, we have the precentral gyrus. That's for voluntary movements. And then behind it, we had what we call the postcentral gyrus. And this postcentral gyrus is where all of the general senses go. So postcentral gyrus for general senses. As we start working our way up through one of these, we'll do fasciculus gracilis. As we start working our way up through that, this will be our final destination. We're going to go out to the cerebral cortex out here along the edge. Now to get to there, we'll have to go through the spinal cord. As we come up, we'll be going through the medulla oblongata. As we go from the medulla, we'll go to the pons, midbrain, and then the thalamus. We'll talk about this as we move along, but this just kind of gives us a reminder of the major structures of the brain. So let's then follow fasciculus gracilis. This is very basic touch, right? So this is for touch and pressure. So let's suppose we have our foot. Let's just do a foot out here. You know, maybe you're putting your foot against, I don't know, maybe you're petting your dog with your foot and you feel this nice cuddly dog against your toes. So here's our receptor. So down here we would have some receptor. And I don't know, there's, you know what, there's Fido. How's that? And you can feel his nice soft fur. We're not dealing with pain, that's the point here. Now to get from here, to get from our foot up to the brain, it will take a total of 3 different neurons. Just 3, that's it. So the first cell, the first neuron is called the primary neuron. Now the primary neuron we would see inside that dorsal root ganglion. So if we looked out here, let me kind of zoom in. Whoops. This would be our receptor. This would be a touch receptor. That'll work. And then this is connected to our primary cell. Right here is the cell body. This is the primary neuron. You'll notice that the primary neuron is here in the dorsal root ganglion. So from that receptor, the sensory comes in, goes straight into the spinal cord. Notice it does not come into the gray matter, but rather it starts to ascend automatically. And this will be going through fasciculus gracilis. So if we were to clear this, let me see if I can't move it out of the way. There we go. So if we were to look here, yeah, we'll change it to green. I don't know why. I always like sensory being green. I wish it was thicker. I wonder if I can change it thicker. Ah, there we go. That right there was fasciculus gracilis. And that's this axon coming up towards the middle of that spinal cord. As we come up, we now get to the medulla oblongata. So here we are in the medulla oblongata. This is where we find the secondary neuron. So here is our medulla oblongata. And now we see neuron number 2 right here. What you'll notice though is that in this second neuron, it actually crosses over. So it starts up on this right side, this is the right side, and then it goes over to the left. So at this medulla, this sensory neuron crosses over. And now it's going to go up to the left side of the thalamus. So from the medulla, we come up here. This is our thalamus. Let me just kind of bring it down. Or I can do it like this. No, it's not going to let me. There we go. I can do it like that. Oh, shoot. My ink doesn't move, though. That's okay. There we go. So up here is your thalamus. And it's in your thalamus where we find the third neuron. And that will then send it to the final part of the brain. So these are the 3 major neurons that we're seeing that, as part of the fasciculus gracilis. Let me go ahead and get out of here. The first cell, the primary neuron, was in the dorsal root ganglion. It goes all the way up through fasciculus gracilis right here. And then it goes to the medulla. In the medulla, we then see the secondary neuron. One thing you'll notice in the secondary neuron is that the fibers cross over, decussate, and then they go up to the thalamus. Up here, we would have the thalamus, and this is where we would find the third neuron. I don't know if you guys remember the thalamus, but we talked about it earlier. It was basically the sensory relay center in the brain. All of the senses going to the brain, except for the sense of smell, passes through the thalamus. So that's where we find the third cell. And then that will relate to the final area of the cerebral cortex. And there you have it, guys. These are the only 3 neurons that we need for the sense of touch. Now, the reason I mentioned that in the medulla, remember this is in the brainstem, we have this crossing over. This will affect different types of lesions, different types of damages. Let's say, for example, you have a lesion in your right lower back, in the spinal cord, and you damage the spinal cord down here on the right side. Let's say we have a damage to this tract right here. Now, ask yourselves, will that affect my right foot or my left foot if it's in the right spinal cord? Actually, we don't need to do right spinal, I'll just say spinal cord. Hopefully you guys will be thinking it will affect the right foot. Spinal cord damage affects the same side. So damage affects same side. However, now let's suppose you come up here and you have some brain damage, and maybe the brain damage is in the right brain over here. Will that still affect the right side? Not if it's above the medulla, because we have this crossing over. Brain damage will actually affect the opposite side because of this crossing over. Yeah. So if I damage my right brain, I will actually lose sense to my left leg. So brain damage, unlike spinal cord damage, will tend to affect the opposite side of the body. So you always have to think about that. You know, what's being affected? Where could these lesions be at? Is it a brain injury? Is it a spinal cord injury? You know, because depending on where you're at, it could either be on one side of the body or the other. All right, so that's it for fasciculus gracilis. With that said, let's go ahead and talk about some descending tracts, unless you guys have any questions. No. All right, descending tracts. Descending tracts, we're talking about all motor, right? This is all motor output. Because we start up here at the top, there's some cells out here in the cerebral cortex, and they will be projecting downwards to either different muscles or glands. Now, the first descending tract that we'll look at is called the corticospinal tract. Corticospinal tract begins in the cerebral cortex. In fact, to be more specific, if you guys remember your brain, here was our central sulcus. Just in front of that, we have this area called the precentral gyrus. I don't know if you guys remember the precentral gyrus. But this was our primary motor area for voluntary movements. So when you decide to move, I decide to raise my hand like this, or I decide to wave to you guys in the camera, that's all beginning in that precentral gyrus. Now, oh, here's a great example. If you wiggle your big toe, you know when you wiggle your big toe? Hopefully you guys remember that it only took 2 neurons to get from your brain down to your big toe. Yeah, that's all starting up here in the precentral gyrus. And this will be going down what we call the corticospinal tract. Now, there's 2 neurons here. There's what we call the upper motor neuron up here. And that goes the entire length of the brain all the way to the spinal cord until you get to the very, very end. So all of this in red is all the upper motor neuron. Then finally, when you get to the level that you're exiting, these cells right here, these are the lower motor neurons. So it only takes 2 neurons instead of 3 for the corticospinal tract. The upper motor neuron in the brain, and then the lower motor neuron in the spinal cord. Now, one thing you'll notice is that the vast majority of the corticospinal tract fibers all cross over in the medulla. And in fact, I might have a picture of this. Do I have a picture? Oh, I don't. Oh, here's a picture. Yeah, on the ventral surface of the medulla, there are these structures here called pyramids. Here's a pyramid, and here's a pyramid. This is where most of the corticospinal tract fibers cross over. I'll show you that in a second. So we have these structures called the medullary pyramids, where we see these fibers cross over. The remaining will eventually cross over in the spinal cord, and that's what we see here, is that most of them come over here, they cross over. Some of them will stay on the front, and then they will eventually cross over later on. The point is they will all eventually cross over. All right, so that's the corticospinal tract. And I'll come back to that in a second. The other tracts that you should get to know would include the reticulospinal tract. Now, one thing you'll notice about these descending tracts is that they're highly scattered. Unlike the ascending tracts, for example, that were all having their areas in one spot, these are just kind of all in a hodgepodge, different areas. So the first tract that we'll see here is the reticulospinal tract. Now, the reticulospinal tract goes through an area in the brain called the reticulum. We'll probably get to that when we start talking more about the brain, if I have time. Anyway, as for the reticulospinal tracts, these are all coming upwards, and they will be conveying information for your sweat glands and to help maintain your muscle tones. So, one thing you'll notice about the reticulospinal tracts, they're just kind of all scattered. There's a few on the side and a few there in the front. The last tract is called the rubrospinal tract. And with the rubrospinal tract, this is actually going to the cerebellum. Actually, it shouldn't be from, it should be to the cerebellum. Oh, no, it is from the cerebellum. My bad, I was getting my ascending and descending mixed up. Yeah. So, from the cerebellum to the spinal cord. So remember when I was talking about moving my fingers and touching my hands like this, how my cerebellum is always monitoring those positions and making those adjustments? That will be going through the rubrospinal tract. That will allow you to coordinate your position, your body movements. All right, so there we have it, the main descending tracts. Corticospinal tracts, reticulospinal tract, and rubrospinal tract. Now, what I'm going to do here is follow one of the tracts down. We're just going to pick out corticospinal tracts. And that's what we see here. And what we can see is that it only contains those two neurons. Here we have the upper motor neuron in the precentral gyrus. So that's the voluntary movement. And it continues all the way down. It goes right through the brain, the brainstem, down to the midbrain. Here's the pons. And then here in the medulla, we have these pyramids. And these pyramids are where the fibers cross over. So as they come down, it will start on the right, or it'll start on the left side, and then it crosses over. Decussation. And now it goes to the opposite side, just like so. When we finally get down here to the level of the spinal cord, then you can see that lower motor neuron right there in the anterior horn. And that anterior horn cell, will then project outwards like this through the ventral root, ultimately going to your skeletal muscle to move it. So lower motor neuron there, everything else is the upper motor neuron. Once again, guys, when we're looking at this, now that we see that they cross over primarily in the medulla, you should start to understand that how this will affect your damage in the central nervous system. For example, if I damage the spinal cord on my right side, or let's say left side, since I've got a picture of the left side. You know, if I affect the left spinal cord, that will affect the muscles over here on the left side. However, if I affect the left brain stem over here, it will actually go to the opposite side. It will go to my right side because all the fibers are crossing over. So once again, guys, brain injury affects the opposite side, the contralateral side. Whereas the spinal cord injury, it will affect the ipsilateral side. All right, guys. I'll tell you what, I think this will be a good place to stop it for the day. Next week, we'll pick it up with the brain. Now that we've covered the spinal cord, the reflexes, I think this will be a good way to end the lecture for the day. So if you have any questions, comments, suggestions, don't hesitate to put them in the chat box to ask. Otherwise, you all have yourself a good week. Oh, I figured that. I thought that might be the case. Y'all have a good week. Yeah, try to get some, try to enjoy the sunshine. That always helps my brain a little bit. You too. I'll see you guys Wednesday. Let me stop my share. There we go.